So what's the difference then between an embryonic stem cell and an adult stem cell? Well, embryonic stem cells are what we call totipotent. Embryonic stem cells have the capacity to generate all of the tissues of the body. In contrast, adult stem cells are what we call multipotent. These cells can generate several tissues, examples such as hematopoietic stem cells of the body that can generate blood, cells of the immune system such as B lymphocytes and T lymphocytes, macrophages, or cells of the hair follicle stem cells. These cells can generate not only hair follicles, but also epidermis and sebaceous glands. There are also cells of the body, stem cells of the body, that are called unipotent stem cells. And these cells have the capacity to generate only one type of tissue. An example of that would be the epidermal stem cell, which, as far as we know, is only capable of generating epidermis. Or liver stem cells, which, as far as we know, are only capable of generating hepatocytes. So, where do embryonic stem cells of the body come from? Well, my laboratory studies the mouse, as do many of the researchers in the stem cell field. And in the early developing embryo, we call this a blastocyst. And already at just only a few days old, the blastocyst already has differentiated into two different cell types. One cell type you see here in pink. These cells are called trophoblast cells. These are feeder cells that support an additional group of cells called the inner cell mass. These inner cell mass cells are the only cells of the trophoblasts that are able to give rise to the fetus. These trophoblast cells are, in contrast, cells that are supporting the inner cell mass. So the inner cell mass of the blastocyst has the capacity to develop into the fetus, and these trophoblast cells are support cells. They do not contribute to the fetus. So another remarkable capacity about embryonic stem cells and about early stages of embryogenesis is that all of this development can happen outside the womb. This can happen in a tissue culture dish. So cells from the inner cell mass can be removed. We can take these cells and we can put them into a tissue culture dish. And now these cells, which become embryonic stem cells, can divide and replicate endlessly. So these cells continue to divide, they continue to generate more and more embryonic stem cells, an infinite supply of embryonic stem cells. And then we can appropriately adjust the tissue culture conditions so that we can regenerate blastocysts in a culture dish. So these embryonic stem cells are capable to give rise to this structure with an inner cell mass and with a trophectoderm. And yet, if we culture our conditions slightly differently, these cells can just divide and endlessly generate more embryonic stem cells. And then, if we take this blastocyst and we implant that into a pregnant female mouse, that blastocyst will give rise to a whole mouse. And so these are truly totipotent cells. We can divide them endlessly in a tissue culture dish, and if we culture them to the blastocyst stage, we can implant those blastocysts into the mice and generate more mice. So, why is this important for regenerative medicine? Well, scientists study mouse so that we can learn more about the biology of stem cells. But this technology has enormous implications for regenerative medicine. How? Well, these cells might be able to be used to generate nerve cells. And nerve cells, if we're able to generate them in culture, could be used to generate or treat Parkinson's disease or Alzheimer's disease, different types of neurodegenerative diseases. Nerve cells might also be useful in treating very bad injuries, spinal cord injuries from football or automobile injuries that could paralyze an individual. If we're able to take these embryonic stem cells and specifically produce pancreatic islet cells, those pancreatic islet cells might be useful for the treatment of diabetes. If we could take these embryonic stem cells and be able to make muscle cells or cardiac muscle cells, we might be able to use them for treatment in muscular dystrophy patients, or alternatively in terms of degenerative heart disorders. These cells might also be able to be used to generate immune cells, which could be used to generate or treat immune deficiency disorders. And finally, as scientists, we might be able to use these stem cells to be able to understand more about the basis of cancers, and that understanding might lead to new and improved treatments of cancers. So for all of these reasons, that's why scientists are so excited about the potential of embryonic stem cells, and we need to know more about the basic biology of stem cells in order to find out whether any of these wonderful, potentially wonderful uses of stem cells might actually become a reality.